Okay, thanks, Vicky. Hello, welcome to the Grand Round. I'm Steve O'Reilly from the Institute of Metabolic Science on the campus, and I'm delighted today to be introducing Professor Arthur Kayser, who's going to present the case and the science himself. When I read his title, I thought it said elections at the center of a storm, but actually, as you can see, I misread that. He's going to be talking to us about electrons. So over to you, and when you're finished, if you can start putting your questions in the chat, I'll read them to Arthur, and we can have a nice discussion afterwards. So, Arthur.
Thank you, Steve. Let me start with two case vignettes. The first one is a 21-year-old woman who had been diagnosed with juvenile idiopathic arthritis at age 14, 7 years ago. And over the last year, she has lost response to etanercept and switched to abatacept. She has also suffered several episodes of anterior uveitis, most recently in October last year, and is on T4 substitution for Hashimoto's. She's now being referred to gastroenterology for increasing abdominal symptoms, which she had actually first noticed at age 10. She experiences right lower quadrant pain and opens her bowel 6-7 times per day with occasional perectal blood and mucus. She also has a moderately elevated fecal calprotectin. The second case is a 20-year-old woman who has been diagnosed with colonic Crohn's disease at age 13. Unusually, she suffers from metastatic Crohn's of her vulva. And over the last 7 years, she has been treated initially with elemental diet, repeated courses of corticosteroids, long-term with azathioprine, albeit with little evidence of clinical improvement. She responded initially to anti-TNF therapy with infliximab, lost response over time due to immunogenicity. Same occurred with adalimumab, another anti-TNF, and she has also sequentially been put on methotrexate, ustekinumab, as well as vedolizumab, and didn't have any response to any of those. She was intermittently severely underweight with a BMI of 15, and MRI earlier last year showed an anterior fistulation into the perivulval tissue. On that background, we decided together with colorectal surgeons for a subtotal colectomy with an ileostomy, which was then performed in March last year. Unfortunately, the vulval affection did not improve. Postoperatively, she was commenced on, yet again, thiopurine and an anti-TNF, and she has also experienced repeated episodes of anterior uveitis and suffers from persistent arthralgia. There are also some neurological symptoms that are sort of non-specific and that we can't really nail yet. Now, both patients are young and suffer from multiple autoimmune autoinflammatory conditions. So non-classical textbook presentation of inflammatory bowel disease is common in very early onset patients where monogenic causes play an outsized role. Now, last time I spoke at Grand Rounds, we featured the phase 2 trial results of risankizumab, the first of a bunch of anti-IL-23 antibodies that currently finish late phase development. And I remain totally convinced that they will be hugely transformative. But they still will not solve the problem entirely. This is already also very clear. And now, despite having anti-IL-12, 23, anti-TNFs, integrin blockers, JAK inhibitors, and you name it, available, we still have patients who are entirely treatment refractory. And this clearly points to disease processes we still don't understand, nor tackle with our current therapeutics. Now, there are precisely two types of human studies that yield data where cause-effect relationships are a priori resolved. One is germline association studies, and the other one are interventional studies in patients, mostly meaning clinical trials. And just the matter to reflect on it is that no matter how sophisticated the high-dimensional data we can generate nowadays is, only these two approaches reveal insight that goes clearly beyond correlation. And indeed, with regard to the first genome-wide association studies have been enormously successful in inflammatory bowel disease. Now we have witnessed an explosion in number of coordinates across the genome that are associated with disease risk in this and many other disease areas. We have now more than 250 individual loci, it's not least thanks to Cambridge faculty such as Miles Parks, who had the foresight in the early 2000s already to assemble large UK-wide and international consortia. Now, the sobering truth, however, is that for very, very few, we have a really true meaningful understanding on how they mechanistically link to disease. And there are yet even some for which we have no biological insight at all, where we do not even understand the gene product's fundamental function. This talk today is about one such locus. Such entirely blank spot was an open reading from chromosome 13. At the time, it didn't even have a proper name. A common valent for isoleucine substitution at amino acid 254, for which about 6% of the global population is homozygous, increased risk for Crohn's disease, as well as for leprosy. And a diminishingly rare loss of function variant, an arginine for cysteine substitution at position 284, caused autosomal recessive monogenic, very early onset Crohn's disease, and the very identical same variant in the same population, indeed, or background, caused Still's disease. Now, Still's disease is often considered the paradigm of autoinflammation with autoimmunity, as it starts in toddlers or young children with a periodic fever syndrome that morphs then over weeks into a very debilitating arthritis. And the range of such variants have now been reported, some causing Still's and some others juvenile idiopathic arthritis without systemic symptoms. In fact, deleterious famine mutations are the sole known cause for monogenic forms of Still's disease. With regard to Crohn's, it's also interesting to note that this puts this gene, famine, actually into the very small but rather illustrious group of genes where monogenic and polygenic inflammatory bowel disease overlaps. And the protein appears pretty old. The C-terminal half is highly conserved with orthologs in bacteria. The structure of several of these bacterial proteins had been solved, and homology mapping produced very good fits for the C-terminus of human famine. Now, the problem was only that there was not a single domain on this molecule that was functionally annotated, or even remotely similar to anything of known function. We used proteomics to survey the molecular vicinity of this protein and found, to our surprise, that the protein sits in a larger complex of fatty acid synthase, the enzyme responsible for all cytoplasmic fatty acid synthesis. Now, famine co-localized with a peroxisome marker, BMP70, albeit without featuring a peroxisomal localization signal itself, and it had previously been described that a sizable fraction of fatty acid synthase and its auxiliary enzymes tethered to the cytoplasmic surface of peroxisomes, where we also found that famine. Now, inactivating famine, indeed, did reduce de novo fatty acid synthesis, but also glycolysis, fatty acid oxidation, and mitochondrial oxidative phosphorylation. Levels of ATP, mitochondrial and total reactive oxygen species, inflammasome activation was impaired, and IL-1 beta production reduced in vitro, and bacterial killing of mycobacteria and salmonella was also impaired. This was all even associated with increased severity of experimental sepsis in an endotoxin model with in fact elevated systemic IL-1 beta levels, and with evidence of disseminated intravascular coagulation. And the big question, of course, was, how does famine do all of that? And based on the structure, we had the reason to believe it's an enzyme. De novo identification of enzyme functions when you then don't have a starting point is really, really hard. So what we what we did is we developed our own method. We identified a cell line that had a non-redundant phenotype upon silencing famine expression. So we assumed that it would contain all substrates and cofactors for its catalytic activity. And ideally, the substrates would accumulate when the protein is silenced. We isolated aqueous metabolites and developed highly quantitative orthogonal liquid chromatography mass spectrometry techniques that allowed us to resolve a whopping 25,000 individual mass spec features. We expressed highly pure recombinant famine in a mammalian expression system and contacted it with this library of 25,000 HepG2 metabolites. And to our delight, we discovered that only very few metabolites changed in abundance. Those substrates whose abundance decreased their potential substrates, those that increased their potential products. Using this approach, we discovered that famine deaminates adenosine into inosine, phosphorylically cleaves adenosine into adenine and ribose 1-phosphate, inosine into hypoxanthine and ribose 1-phosphate, guanosine into guanine and ribose 1-phosphate, and methylthioadenosine into adenine and methylthio-ribose 1-phosphate. Now, these reactions have been thought to be the domain of ADA, PNP, and MTAP. Whilst this reaction here was considered outrightly absent from eukaryotic metabolism. It's also worth noting that a deamination reaction has very different structural constraints compared to a phosphorylitic cleavage. Famine's catalytic activities were indeed evolutionarily highly conserved, as we also found it in its bacterial orthologs. And I don't need to remind anybody here that adenyl as well as guanyl groups are the center of the major energy currencies of a cell, and adenyl groups are the defining feature of all of the major cofactors. Now, adenine, adenosine, and ribose are also considered primordial metabolites from which life has emerged from prebiotic chemistry billions of years ago. And central purine metabolism is therefore, perhaps unsurprisingly, conserved in any form of life. Now, since de novo purine synthesis leads straight to inosine monophosphate, the generation of the purine nucleosides, as well as the nucleobases, was thought to be the sole domain of ADA, BNP, and MTAP. However, now it turns out that adenosine and adenine can be directly interconverted also in eukaryotic cells, and the monofunctional enzymes are not alone here either. One would conclude that there is redundancy, but loss of function of the enzyme tells us differently. Now, MTAP is deleted in about a third of all cancers and thought to account for the tumor cells' altered methionine metabolism. ADA and BNP deficiency causes severe combined immunodeficiency. It is characterized by a complete lack of T and B lymphocytes. Now, a compromised activity of famine instead resulted in autoinflammation and autoimmunity. ADA, BNP, and MTAP are ubiquitously expressed, so how could this be? It's also worth noting that the 254V variant is not just quantitatively impaired, whereas the 254I variant cleaves adenosine into adenine. The 254V variant has a preponderance for deamination of adenosine into inosine and onward metabolization to hypoxanthine. Now, macrophages from mice genome edited to express the human SNP variants showed a gradual decline in adenine, adenosine, and ATP levels from 254I to 254V to 284R. And to make a long story short, what underpinned these observations, these metabolic consequences, was that famine enabled a pure nucleotide cycle. When famine was less active, such as in the V variant, the cycle became slower, and it came to a complete standstill when famine activity was absent. Now, a pure nucleotide cycle had originally been proposed by Löwenstein and Dornheim at Brandeis in the early 70s. A cycle deaminates AMP to IMP and then reaminates IMP via succinyl AMP back to AMP. The nitrogen for the reamination stems from aspartate. It's only consumed substrate, and aspartate's carbons are then released as fumarate. Now, Löwenstein and Thornham had brilliantly demonstrated this in extracts of skeletal muscle. It's really great literature to read. They had this science paper in 1971, and then almost every year JBC papers working up this mechanism, all in skeletal muscle as extracts. Now, outside skeletal muscle and even in skeletal muscle, the cycle has remained elusive and obscure and is therefore mostly ignored. Without showing you any of the trace and perturbation data that supports this model, what farming does in macrophages is that it controls carbon flux via the PNC, thereby pulling citrate carbons via ATP citrate lyase, ACLY, into oxaloacetate, into aspartate that the PNC consumes, and aspartate's carbons are then released as the fumarate, the mentioned already, which is hydrated to malate. And via this cycle, farming balances electron flux from glycolysis into mitochondria via the glycerol 3 phosphate shuttle, as well as the malate aspartate shuttle. Coming back to the disease association. Now, about 20% of children with Still's disease develop hemophagocytic lymphohistiocytosis, also known as macrophage activation syndrome. It is typically triggered by trivial viral infections and has a horrific mortality rate. In HLH mass, CD4 and CD8 T lymphocytes expand quite massively, and this T cell derived interferon gamma that is being secreted appears to activate macrophages to become hemophagocytic and to release large amounts then of inflammatory cytokines. It's poorly mechanistically understood overall, and HLH mass has also been implicated in mortality regardless of Still's disease from viral infections and many other conditions actually, including influenza A virus infection. And so with farming the sole known gene causing monogenic forms of Still's disease, we recently had an ideal model to study HLH mass predisposition. We infected mice with H3N2 influenza A virus, and those with absent farming activity fared by far the worst. They exhibited increased tissue damage in the lung, and they also had a marked increase in plasma interferon gamma, as well as IL-10. This is interesting since influenza A virus-specific T lymphocytes characteristically co-express interferon gamma alongside anti-inflammatory IL-10. Using tetramers, we therefore quantified influenza-specific influenza nucleoprotein specific T cells, and quite remarkably, they were already 4 times more abundant in the common polymorphic 254V variant compared to 541I, and yet higher in the 284R that outrightly lack farming activity. And it's well known that T cell responses can drive lung immunopathology in influenza infection. It's also known that they require lung dendritic cells for their activation. So we deleted farming specifically and selectively in dendritic cells only, and yet again the influenza-specific T cell responses in the bronchoalveolar lavage doubled. This raised the question on whether farming controls T cell priming by dendritic cells. Now, T cell priming requires antigen presentation by MHC recognized by a T cell receptor. It requires co-stimulation and signals from secreted factors such as cytokines. Now, farming is highest expressed by far in dendritic cells and macrophages. T lymphocytes, in contrast, especially naive T cells, do not express farming. Now, to study this, we used ovalbumin as a model antigen and CD8 positive OT1 T cells with a transgenic T cell receptor that recognizes the relevant ovalbumin peptide. Now, farming deficient dendritic cells pulsed with ovalbumin primed for much more T cell proliferation and interferon gamma secretion than dendritic cells with intact farming. These T cells were also much more cytotoxic than those primed with intact farming, and they released much more granzyme B from their cytotoxic granules. They also secrete massively more interferon gamma, as you can see here, upon the re-stimulation. Increased priming by famine-deficient dendritic cells extended to CD4 positive T cell responses, and therefore, to summarize here, reduced or absent catalytic activity of famine within dendritic cells resulted in a profound stepwise increase in the priming of CD4 and CD8 positive T lymphocytes, which was associated with lung immunopathology. Despite all this, the transcriptomes of dendritic cells with and without famine activity were identical. There was no transcriptomic footprint at all. Now, CCDC 122, the only other transcript with differential abundance, apart from famine itself, sits genomically right next to famine, and it appears to be affected by the gene trap here. We also compared, for example, 254I and 254B dendritic cells, and you remember I have fourfold more influenza-specific T cells that they primed for. There was not a single transcript that was differentially expressed. Now, this absence of a transcriptional change was not entirely surprising to us, since we had observed precisely the same in macrophages. And we also looked at the protein expression in dendritic cells. There was no difference in co-stimulatory, co-inhibitory molecules, nor in secreted cytokines, such as IL-12 or interferon alpha. So how could dendritic cells become pathological primers without invoking transcriptional activation? We noticed an increased pace in antigen uptake, measured here with a fluorescently labeled model antigen. This was also linked to increased pace of antigen transfer from the endocytic compartment to the cytosol, and such transfer is a prerequisite, as you know, for cross-presentation in MHC class 1. And indeed, this was also reflected in faster arrival of cognate overpeptides on MHC1 at the cell surface. It turned out that at the basis of increased antigen presentation was the interconversion cycle, or the interconversion cycles of adenine and guanine nucleotides. When famine is fully active, the operation of these two cycles is well balanced. When famine catalysis is impaired, the lower part, the pure nucleotide cycle itself, slows, while the IMP, XMP, GMP cycle increases in pace. Now, IMP dehydrogenase and GMP synthase, IMPDH and GMPS here, are hyperactive, which leads to increased reduction of NAD plus to NADH, shifting the cytoplasmic redox ratio. When famine is catalytically dead, the BNC comes to a standstill, whilst the IMP-XMP cycle is even more active, generating cytoplasmic reductive stress. And it's important, as we discovered, that this increased membrane traffic and therapy thereby increases antigen uptake. And the key experiment that supports this conclusion comes in the next slide. Pyruvate can regenerate cytoplasmic NADH to NAD plus via the LDH reaction. Pyruvate indeed rescued the increased antigen uptake in dendritic cells with inactive famine, whilst it did not impact on those with active famine. Now, since pyruvate is also mitochondrially metabolized, of course, this does not prove that NADH reoxidation was responsible for the rescue. Now, alpha-ketobutyrate is an alternative substrate of LDH. Neither itself nor its product, and we've confirmed this in dendritic cells, is further metabolized. And alpha-ketobutyrate indeed phencopied the rescue achieved by pyruvate. This demonstrated that cytoplasmic reductive stress results in enhanced antigen uptake and presentation in dendritic cells. Now, IMPDH is also the target of mycophenolate. And mycophenolate also rescued enhanced antigen uptake in dendritic cells that lacked famine activity. This provides, I think, a pretty new perspective on this immunosuppressant's mechanism of action. There was one more thing, quickly. We noticed that in addition to enhanced antigen uptake and presentation, there might also be a soluble factor involved. When we took supernatant from dendritic cells and transferred them to naive CD4 or CD8 positive T cells that were stimulated with anti-CD3 and anti-CD28 antibodies, we observed a marked increase in their interferon gamma secretion when the supernatants derived from famine-deficient dendritic cells. This difference in their ability to induce interferon gamma was retained when we filtered the supernatants via a 3 kilodalton filter prior to putting them onto T cells, so also pointing toward a small molecule. The difference in the activity of dendritic cell supernatant was particularly stark between genotypes 254I and 254V and extended beyond the different gamma induction to T-cell proliferation, as you can see here. So the difference essentially here in proliferation is really quite remarkable. Now, amongst more than 1,000 LCMS features,
you'll need to wind up.
I'm almost finished. I'm almost finished. Amongst the 1,000 LCMS features resolved within the dendritic cell supernatant, inosine was the top-ranking identifiable LCMS feature. It was only one of 2 metabolites that overlapped with a second screen. It was a very plausible candidate, of course, and indeed dose-dependently decreased proliferation of naive CD3, CD28 stimulated T lymphocytes. This suggests that inosine provided an inhibitory signal, and this is what we could actually show by agonist as well as antagonist treatment here. So to summarize, we have discovered a purely biochemical mechanism that restrains T-cell priming by dendritic cells. It involves a redox-based control of antigen uptake and presentation and the release of inosine. And this mechanism of restraint is defective due to reduced catalysis. T-cell priming is markedly enhanced and can result in a systemic cytokine storm and profound immunopathology in the lung when it's completely absent. Final point, this enhanced priming activity may have brought evolutionary benefits. This paper by Rivas sought to identify genetic risk factors of Crohn's disease that may account for the markedly increased risk for Crohn's disease in the Ashkenazi Jewish population compared to the Caucasian population. And the I254V polymorphism is, together with an IgM polymorphism, the largest contributor here, even larger than NOD2, which has been linked to a founder effect. The most important point, though, is that digging deeply into a disease risk locus revealed here an enzyme that operates at the very center of energy metabolism and is conserved from bacteria to man. And with this, I'd like to close. Science Great Team Sport. I'd like to thank current and former members of our group, as well as our absolutely terrific and wonderful collaborators. Discovery of thiamin's catalytic activity was driven by Saim Kader, who is now in the Wellcome Fellowship at Cornell, and starting the work really from a blank sheet of paper during his PhD, together with James West, who runs our mass spec unit, and Rodriguez, who share joint credit. The immunology story that I shared with you was with enormous enthusiasm driven by Svetlana Savelyeva, Gavin Sewell, and Katarina Ramsvan, who share joint credit and who has always received a lot of input from everybody in the group and from our collaborators. In that regard, I'd like to mention in particular Yogo Moses, who've helped with the structural work. Sadly, our dear friend Rodrigo unexpectedly passed away just 2 weeks ago. Having completed his PhD in early 2020, he secured a postdoc position with Jeff Pollard in Edinburgh. Was very proud of that, and his scientific contributions will undoubtedly live on, but we sorely miss him. Thank you for your attention.
Thank you, Arthur, for that fascinating science. Unfortunately, you've left us only with about a minute to ask questions. Patrick Maxwell has congratulated you on the fascinating story. Does it tell us anything about the intersection between diet immunity and BMI and immunity?
We don't know yet. Actually, we have started to investigate this, but I can't give you a firm answer here. What is interesting in this context is that the mechanism essentially that has been linked to the probably most promiscuous GWAS signal in glucokinase regulatory protein seems to invoke precisely a similar mechanism of reductive stress in hepatocytes. And we think essentially it may be that this is a major link how essentially obesity is also linked to autoimmunity.
Okay. One question from me quickly. This gene is expressed widely. Why is its pathology so limited in a way to the immune system or the obvious pathology, and not, for example, in the CNS where it's highly expressed?
Great question. We're investigating this, especially CNS expression is pretty intriguing. We have a few ideas essentially why this is particularly interesting and relevant. But this requires further work. Yeah.
And then
myeloid cells really stand out actually for particularly high expression actually.
Okay. In terms of just time, we just have time for one last question. There's several in the chat. Please have a look, Arthur, and answer people afterwards maybe. But John Firth has asked, apart from the use of MMF, does this understanding suggest any new approaches to treatment of this particular group of patients?
Yes, I think so. I mean, there may be essentially an aspect that is related to personalization of treatment, and I think it gives a completely new perspective, not only on mycophenolate, but also on a few other small molecule immunosuppressants that we're investigating this.
Okay. So thanks. Please have a look at the chat and answer some of the questions of the people who asked them. We need to move on to the second presentation, which is from the Department of Surgery, and we have Ms. Ekpemi Irune, giving us a presentation on the surgical metaverse, mixed reality technology in surgical care. And Ekpeni, you may want to instruct us on what we need to do other than being on Zoom. I think there's some...
Hi, hello. Yes, hello everyone. Thanks for having me. And thanks to Prof. McCaskey for asking me to do this as well. I've got two colleagues that are also joining me. And because this is a call that's using several interfaces, we're using Teams, we're using Zoom, and we're also going to be using a few HoloLens devices. Please bear with us as sometimes things take time and there may be some technical glitches, but hopefully it's going to be fine. I was going to send you all a Teams invitation in case anyone wanted to have this more interactive, but I will start the process just as we are, but if you are going to log into Teams, you would need to be on a desktop. But for now, we're going to keep everyone on Zoom for simplicity. But if
you're not sharing your screen at the moment, we're just seeing
yeah,
your
not
photo.
yet. So I'm going to share the screen. Yeah, right. So here we go. I'm going to share that now. And so what we have is this is my Teams screen. And I'm joined here by two fantastic colleagues of mine. The first person that's here is a gentleman called Phil Moore, who is Digital Innovation Lead at for public sector from Insight. Hi, Phil. You can wave. And Phil is actually, where are you based at the moment?
In Hampshire.
He's in Hampshire. And I've got Yassine Baradi, who is a mixed reality consultant. And where are you, Yassine?
I'm based in Paris, France.
Fantastic. So my colleagues, I hope you can all see this and see the screens. And all three of us have got a HoloLens device on. And this is a device that allows us to access a virtual reality world. But we are on the screen together. We're in this environment together because of a tool known as Remote Assist Dynamic 365. And this creates the virtual environment where we can link in with each other from anywhere we are in the world and share a physical space, exchange information, annotate, train, learn. And this is the basis of this, the beauty of this sort of technology. So you can imagine already the kind of use cases that you can apply this technology to. And Phil is just having a play there with my screen. He's sitting in his own home and just having a play with my screen. But we can be very, very interactive with one another. So virtual reality is one of the components of mixed reality, the other being augmented reality. Augmented reality allows you to augment the physical world using computerized tools, software applications, etc. And you can access that on your smartphone or a computer device. Virtual reality, on the other hand, allows you to only augment the artificial world. So, and you see examples of that as we move on. And when you bring the two together and other components, we refer to this as mixed reality. So we're going to go through, because we have a lot of things to show you, and we're just showing you basic things of the potential uses of this. And I'm going to ask my colleague, Phil Moore, to just start this session by showing us an example of a three-dimensional interactive hologram, in particular the coronavirus. So what the beauty of this is that you can see Phil's own eye perspective, so from his view. And this is him showing us the coronavirus. He's going into the virtual world now. You can see how futuristic it looks. Great. And that's the coronavirus. And you can see he can move it around in his room. Can spin it around. And if you were in the same room with him, you would be able to walk around it and look at him twisting it around. And I think he can even split it. Can you? Let's see. And there we go. And you can see the different components, the S proteins, the RNA in it. And that's a very nice way of showing someone or describing something to someone or teaching someone and giving them an idea. You can imagine describing those receptors, the surface receptors, and people not really understanding how three-dimensional they are. And this allows you to be able to really get a feel for it. Of course, a lot of companies work developing these applications. And right now, this sort of technology is being implemented across the board in the National Health Service as well. Great. So I think my turn now. So I'm going to pop my HoloLens on and hopefully you can now see what I can see. Hopefully, I'm going to get up. Can you see, Phil, have you got me on?
Yeah, you need to check, focus on your screen, I think. Amy.
Right.
Because it's showing me on the main screen at the moment.
Okay, yeah, all right, so that's me. And you mean in Zoom, right?
Yeah, well, I'm only seeing you in Teams, and I'm just seeing I don't know if it's just my local view.
Yeah, I think, well, I think, let's just see where we are. I'm on Zoom now, so I'm just going to
just check to see what view they're seeing on Zoom. Are they seeing your view as the main image?
No, they're still seeing you. But if you mute your mic, it might help. Let's try that.
While we're doing that, I'll just get up the COVID patient just in case we can't get that going.
So I think everyone can hear you on Zoom. So if you maybe mute your mic, I'm going to try and see if I can make myself the presenter. Yeah, I'm now the presenter. That's perfect. So, right. Hi, everyone. Hopefully you can see me and you can see I'm just going to move my screen a little bit over to one side. And that's me just moving my screen. Okay, good. Just move over here. And so you can see what I can see, hopefully. And I'm going to go into apps, all apps. And what am I showing them now? The holoanatomy, isn't it? So we're going to go into the holoanatomy. And it's just loading, so you should be able to see nicely. And again, I'm just going into the virtual environment. And hopefully you can see that. And you can see it's just opening something called holoanatomy. There we go. All right. And you can just see that. I'm just going to drop that. Here, actually. Good. I hope you can see that. You have a good view of that. So we're just going to go into demo 1 and start session. Okay. And hopefully you can all see this. It's just going to prepare a little slideshow and you can see that's there. I'm just going to see if I can, and you can see there that it will ask me now about doing something else. And you can see it puts the muscles on. And you can imagine the instructor describing all of this structures and talking about, and I can walk around it, close to it. I can also see if we can do something else with it. Good. Let's go back here. And let's a bit of some muscles on it as well. And a little bit more, kind of pulling back the muscles there. And you can see the inside there. And you can see the different things. You can look very closely at all that and look at all the organs, etc. And sometimes you can even twist the entire thing and walk around it. Good. So I'm glad you can see that. Good. And I think the next thing we're going to move on to quickly because we don't have enough time. You can make that bigger and bigger. It looks massive now. And this is expanded by over 200%. And here, this is the nice part of it, being able to literally go under it and look up. I can come over here and look at it really nice and closely, right? And I can look inside bits of it, and it just keeps going. I can see inside the intestine there and the gallbladder. Great. Still remember some anatomy, so that's nice. Good. So I'm going to now come out of here, and we're done with this. A little bit more information there, but I think you get the general gist of this. Good. All right. So let's get out of here. And I'm going to go back now and show you about the hollow patient. And we're going to use something called the hollow patient, or we're going to use the COVID-19 patient. And that's actually quite a nice one because it's pretty topical. And again, it allows you to be able to move inside the virtual world. And yep, we're going to press yes to that. So this is a scenario here that's telling us, we can imagine teaching a patient, teaching students using this. This is a scenario-based learning, and it tells us here, welcome to the hollow patient COVID-19 edition, and we'll be looking at signs and symptoms typical of COVID-19, assessment, risk stratification, and press or say start to continue. So I could say start, or I could just press start. Good. And then that's my patient. So I can move my patient closer to me. Can you see him? Good. All right. And then we're going to say done. Okay. And so this is a patient. He's male, comorbidities, hypertension, 59 years old. And he's coughing, isn't he? He's on amlodipine, and he began feeling ill some time ago. And like I said, you can actually walk around the patient and have a look at him like that. He's sitting on a stool. You can't see all of the stool, but it gives you a really good view, doesn't it? And here we can go through the scenario and he says feeling ill, and it all tends to is become even more ill, and now he's on oxygen. They've taken the COVID-19 swabs. He's coughing a lot. I think he might actually pass this thing on to me. I have no mask on. Okay, breathless increasing, and of course, we'll just keep going forward. And then finally, God, he's getting really bad, isn't he? We have no ITU beds, I don't think. But again, you're getting more information. So you can imagine how you can interact with a lot of students or trainees and take them through the scenario in real time. And those people who have never seen a patient get really unwell can get an idea of the kind of signs to look out for. And finally, I think here, yeah, he's got some non-invasive ventilation on. Pretty poorly, isn't he? And they've started CPAP. And this scenario takes you all the way to him. And I think the last scenario shows that luckily, I think he might have gotten better with this. So we're going to, and you can see over there as well, you can see the blood pressure and ECG trace, heart rates, etc., and temperature. And you can imagine how you can customize and write any scenario to teach anyone. And these scenarios, people like Insight, for whom Phil Moore and Yazin work for, can help us create these scenarios to train anyone to do almost anything. Now we're going to come out of here and get a little bit more technical. Phil, is the next one guides?
Yeah, you'll need to do guides, because my iris tracking isn't working.
That's fine. So finally, we're going to get a little bit more technical and go into guides, because all we've done so far is a little bit more clinical, patient-facing kinds of things. So you can also imagine that if you were on a ward round and you needed a consultant review, you can bring everybody into the patient's room and they can see your own viewpoint, and your specialist can be anywhere in the world. So let's go into guides. Now, guides is more about procedures. And I think this is where the HoloLens really comes into its own. And HoloLens is not just used in the medical field. In fact, it's used in a lot of building and construction, in a lot of technical fields, aviation industry, etc. And it's used for a lot of training. And so I'm going to show you an example. We're just going to try a guide, but we're not using a medical guide. This is just a technical guide on how an industrial or an organization is going to train people how to do something. So look at that. I don't know if you can see that now. Phil, I don't think they can see my screen.
I can see your screen here. I can see yours as well.
So I think they can only see me at the moment.
Maybe the focus is on your teams and not on your HoloLens.
The focus?
Yeah, on Zoom. On Zoom, maybe the focus is on your teams and not on your HoloLens, I guess.
Oh, hold on. Let me. I don't know what's happened here. Let's see if we can. I'm just going to stop the sharing and start the sharing again. Seem to have come off of there. There we go. Let's share that again. Let me see if that works. So no, I think it's still on my, I think we've lost the.
I can still see your.
Yeah, that's because you guys are all in HoloLens.
Yeah.
But I think
Yeah,
that.
it's yours as well.
Yeah. I don't know what's happened there. I'm sharing my team's screen, but that's. So I'm sharing my team's screen, but they can't see my team screen. They can only see my HoloLens screen. Someone's written. Just gonna have. There we go. All right, we're good. I think it's just the focus that needed to be adjusted. So let's get out of here. All right, so now this is guides. And guides is great because look at that. There is a little information board somewhere. So this person is learning how to do something. And I can actually use my eyes to do everything. I'm not using my hands, and I can say, confirm to proceed. And all I have to do is look at it. And that's it. It's done it. So you're all set. And this is the virtual workspace is trying to train me how to use. And it's trying to show me what to do. So if I want to go next, I just look at that arrow, again, hands-free. And it tells me what I need to do. And again, let's get out of here. Next slide. And it keeps telling me how great it is. Let's move on. Good. And we're just going to do guides overview. Good. All right, so this is a task and it's telling me what to do. It's telling me to pick this up. Yeah, and it will tell you exactly what to do. So imagine trying to learn. So pointing to a specific location is what it does. So if you want to teach someone, for example, how to place a cannula, you can imagine using this. If you want to teach them how to use a ventilator, you can use guides to do that. If you want to tell them how to calibrate a piece of equipment, you use guides. And people can do this in the comfort of their own offices, or if you had a room or a facility where people can go and do any course at all. And actually there's an organization that's even using this to get people to recertificate for basic resuscitation and advanced resuscitation, etc. So it means you don't have to wait for a slot to open up in real life. You can just go whenever you want, and you can follow the instructions. Good. So at this point, I'm going to stop because we've shown you quite a lot. Phil, are we done?
I think we're done, yes.
Yeah, brilliant. And the time has passed by. I'm going to take my HoloLens off now and stop sharing the screen and then chat to you all. So if anyone has, the only thing about HoloLens is that you then get hat hair, but that's all right. We're used to that as surgeons. So I hope that was self-explanatory. You know, if the HoloLens works intuitively more with the Teams, and so it's like joining a Teams meeting when you use the HoloLens. And also that, of course, has an impact on costs. It's the cost of a phone call, probably less. And also, most importantly, is that in the NHS, you know, we're rolling out Teams for everyone through the Azure client, and you know, Microsoft Windows is the operating system of the NHS. So right now we have some good news because we're able to access to get a fund. I was able to get a fund of a £250,000 that's coming into our Addenbrooke's Hospital in order to implement how we use some of these devices, and so that's good news. And my colleagues Phil Moore and Yazin will also will be helping us develop some of these use cases as well. So I'd like you guys to thank them for their support and their assistance, and I'm happy to take any questions.
Thank you, Ms. Renee, for making your way through the challenges. I'm sure the Lumiere brothers had a similar challenge when they first showed their moving pictures. Can I just ask you, you've emphasized the educational issues of the potential bonuses of using this sort of technology. I mean, you're a surgeon. I think we were kind of expecting to hear maybe some examples of how it actually directly impacts on patient care. Do you think the technology is getting to the stage of actually having impacts on surgical practice?
Absolutely. I mean, apart from, you can use this to train. So for example, even from the basics, ward rounds. I don't necessarily have to be present on a ward round. If I'm somewhere else, I can dial in remotely and I can see what's happening with my with the trainees as they do a ward round. That's something you can do. We certainly also delivering care. If someone is seeing a patient in another part of the hospital, I'll be able to dial in and see this. If I was going to take someone, for example, through how to do a thyroidectomy, for example, it will make sense for them to start with something with the virtual environment where they can see something, they can see the three-dimensional anatomy and appreciate what the procedure entails. You can use guides to create a step-by-step approach to doing a thyroidectomy. You can supervise them remotely for the first time without you being in theater. And so there are lots of inputs in that situation for surgical training. And then when even thinking about the current situation we are with the pandemic, there were a number of hospitals in the NHS that used this to be able to review remotely patients who were isolated or quarantined. And then there's that community care as well. And then collaboratively, if I'm operating here in Addenbrooke's, I can put my HoloLens on. It doesn't obstruct my line of sight. And any of my colleagues anywhere in the world can dial in and, proctor me on doing a new procedure, for example, or something complex and have inputs. And so that's the beauty. It democratizes access to technology, to skill, to expertise. And this is the future.
There's a question from OS. I'm not sure who OS is, but they've asked, what level of customization would you expect to be available, and would clinicians be able to do this with ease, or best left to IT technicians? And for which use scenario are you most excited about?
Well, I'm sure my colleagues and I are excited about the ward round ones, because it would be nice to be able to, to be there for cases that you need to do without necessarily being physically present. For example, I'm also excited about what we could potentially do with imaging during live surgery, and superimposing cross-sectional imaging on patients that have tumors, etc., and using landmarks and being able to find those tumors much more quicker with smaller incision, etc. Those are the kind of things that I'm excited about. With regards to use, it's very easy to use. You don't need any, it's very easy. It's a tool that you pick up, and very soon, I think in the next few years, we'll have a number of them within the hospital. But when it comes to developing what you want to use it for, you need experts to come in and help and support that. And that's why we have people like Phil. But eventually, I think the way forward is that the NHS within IT will have teams like this embedded, because it will now be standard of care. It will be a situation of, you want to develop training or a clinical scenario or support in clinical care, and you would have teams within your IT department that will be able to help you write and craft the scenarios and bring them to life, quality assess them, etc. The other thing just to mention is that our postgraduate medical education led by Aaron Gupta is already light years ahead. They've purchased 10 HoloLens, and I think that they did that with the help of Jonathan Morton a couple of years ago. And they have a huge amount of money to spend on digital transformation, and there's a simulation center that's also planned. So watch the space. We're very lucky in Adam Brooks to have people who believe in this technology.
Okay, another question from Carol Davis-Wilkie asks, can't we use the normal functionality, say like Zoom, for ward rounds right now without needing a holographic headset?
Well, it's no comparison, because with Zoom, you're going to have to, how do you focus the camera? You're going to take a laptop and point it at the patient. From someone who's used these devices in different forms, the HoloLens, there's no comparison. I can look right into someone's mouth with my HoloLens device, and my consultant can, and my colleagues can see what I'm seeing in living color. And so I think it's a very different situation. It also makes sure that your hands free, and therefore people have your own line of sight. And that's the most important thing, that line of sight. And then the collaborative aspect of that, and being able to bring that person into your own world, where that person can annotate your screen, your live site. I mean, you can't do that you can annotate the Zoom screen, but it's very different when the person is saying, that's the peritonsillar abscess, stick the needle in there.
So there's a lot of concern at the moment that artificial intelligence and other technologies will take over from humanity. Are your educationalist colleagues worried that what they're doing, all that money we're pouring in, is going to put them out of a job?
I think you would always need human beings to manage artificial intelligence, because they're not completely autonomous. People need to write the programs, people need to manage them, and provide oversights. The tools and applications need to be supported. I think we have to be broad-minded and just think about diversifying and adapting and evolving.
So
yes,
we won't get cheap medical schools that we can run for 10 quid a year.
Exactly. Look at that. Yeah, absolutely. It allows it chip-ins everything. It makes it more accessible.
Thank you very much. It's been a fascinating insight into this new arena, and thank everyone for their questions and participation today. Okay, cheerio.
Thank you. Bye.
Bye.